Hello and welcome everyone. Sonia Pellici and myself are very pleased and proud to present you this symposium on the theme of visceral brain axis and mental health. I will be brief in order to leave as much time as possible to the presenters to expose their exciting findings. So in few words, why this symposium? Because it is largely admitted that body and brain have continuous interaction, in particular via the autonomic nervous system. The ANS is widely known to be involved in homeostasis, physiological homeostasis, of course, but also cognitive and emotional homeostasis. The ANS allows quick reaction to internal and external modification, and it's like a superhighway carrying information between the brain and the internal organs in both directions. The objective of this symposium is then to present recent work that have assessed how body activity in link with the vagus nerve, namely heart rate variability and gastric activity, is related to normal and pathological functioning. The presenters have investigated whether and how the viscera and brain are able to communicate in a bidirectional manner. Findings for several approaches will be presented in order to better understand emotion-cognitive interaction in HRV and gastric activity. Additionally, we know that numerous fibers of the ANS are afferent, mediating information about pressure, tension, temperature, brain, and so on. For example, in the vagus nerve, more than 88% of the fiber project toward the brain. The vagus nerves tell the brains what's going in your organs, more especially the heart and the digestive tract. It is then expected that signals for our body are integrated in brain and influence our mind, thought, emotion, and ultimately our behavior and your relation with others and the environment. A part of this talk will examine whether the afferent information have influences on the cognitive functioning. At last, the last years have seen an important craze for techniques aiming at modulating the activity of the ANS in order to improve the efficiency of cognitive function, such as voluntary slow breathing. Talk of this symposium will then also assess effect of such techniques on both normal and pathological functioning. So we wish you a good symposium.
The title of my presentation is, Why Should Psychotherapists Care About the Vagus Nerve? A Neurovisceral Integration Perspective. I want to cut to the chase and give you the punchline first. In 2018, we published a paper on pre-treatment heart rate variability, predicting treatment dropout and residual symptoms after exposure therapy in patients with panic disorder and agoraphobia. What we were able to show, this is a slide of the participants. 28 recovered, 161 had residual symptoms, and 38 were treatment dropouts. And this is just some of the demographics of those participants. Importantly, what we were able to show was that there was a relationship between heart rate variability pre-treatment and therapy outcome. Specifically, those individuals that had residual symptoms or dropped out had lower heart rate variability pre-treatment compared to those that fully recovered. Importantly, when we looked at the treatment dropouts, those individuals that dropped out for motivational reasons, such as too much fear to enter the exposure situation, had lower heart rate variability compared to those that dropped out for other reasons, such as having moved away from the treatment location. So, heart rate variability may be important for psychotherapists as a predictor of treatment outcome, and heart rate variability may be a measure of vagus nerve activity. So, psychotherapists might find the vagus interesting in their work. This is just the logistic regression for the residual symptoms, the results highlighted for heart rate variability, and for the treatment dropout. So why the vagus? Darwin, in his book on expression of emotion in man and animals, talks about the work of the French physiologist Claude Bernard. And he notes, Claude Bernard also repeatedly insists, and this deserves a special notice, that when the heart is affected, it reacts on the brain, and the state of the brain again reacts to the pneumogastric nerve on the heart, so that under any excitement, there'll be much mutual action and reaction between these, the two most important organs of the body. So in the mid-1800s, Claude Bernard and Darwin were already talking about the relationship between the heart and the brain via the vagus nerve. To me, this is fascinating because of what was known at that time about the heart, the brain, and nerves in general. Just amazing. So this is an overview of the autonomic nervous system showing the sympathetic division and the parasympathetic division. The vagus nerve is the major conduit for the parasympathetic division. The vagus nerve is one of the longest nerves in the body and goes all over. And in fact, the word vagus is derived from the same word as vagabond or wanderer. So the vagus is often referred to as the wandering nerve. This is another figure showing the vagus nerve, and it highlights the fact that there's a left vagus and a right vagus. And it also highlights the fact that via the nucleus tractus solitarius, the vagus goes into the central nervous system and the prefrontal cortex. So based on this information, we had proposed some years ago the neurovisceral integration model, suggesting that there is a network of neural structures that can be indexed by heart rate variability, which is a surrogate for vagus nerve activity, and that higher heart rate variability was associated with greater prefrontal inhibitory tone. Importantly, a lack of inhibition leads to undifferentiated threat responses to environmental challenges, and that this lack of recognition of safety may be at the heart of psychopathology. This is a figure from Eduardo Benarroz showing the central autonomic network. There is bidirectional communication throughout the neuraxis, but importantly for our discussion, the output of this set of neural structures, which includes cortical structures, midbrain structures, medullary level structures, is via the stellate ganglia and the vagus nerve to the sinoatrial node of the heart. At the heart, it produces time interval timing differences, which represent heart rate variability. And this figure shows in the blue the fast variability, and in the red a slower variability. This fast variability in the blue is almost exclusively due to vagal activity. Heart rate variability can be measured in the time domain or in the frequency domain, and this high frequency vagal activity is represented in the frequency domain in this area in yellow. That's the primary focus of the rest of our presentation today. This figure from a 2005 paper in rodents shows the relationship between high frequency heart rate variability, as we might assess in non-invasively in humans, and vagus nerve activity measured invasively in these rodents. And what it shows is that there's a strong association between vagus nerve activity measured directly and high frequency heart rate variability, suggesting that our measures of heart rate variability might be a useful surrogate for vagus nerve activity. In 2012, we published a meta-analysis on heart rate variability in neuroimaging studies to further quantify this association between the heart and the brain via the vagus nerve as implied by Darwin. What we found in this meta-analysis in the yellow are the areas across task and across modalities that were associated with heart rate variability. And in particular, we identified a pregenial anterior cingulate and a subgenial anterior cingulate region, primarily on the right-hand side, and the left amygdala. Now, these prefrontal regions have been characterized, and this is a characterization from Amodio and Frith, showing that these regions are associated with emotion and social cognition and reward, punishment, and outcome monitoring, all important aspects of psychological health, but importantly, also sites of visceral regulation. So over the years, we've looked at heart rate variability in organism health and shown heart rate variability to be associated with physiological health, such as hypertension, emotional health, attentional processes, and cognitive health. These latter three are particularly germane to our topic today. In 2015, my colleague Ted Bouchain and I published a paper where we proposed that heart rate variability might be a transdiagnostic marker for psychopathology. This is based on the work of Caspi and colleagues that identified a P factor, a general psychopathology factor in the structure of psychiatric disorders. Early characterizations of the latent structure of psychopathology identified, for example, by this paper from Kruger in 1999, suggested that individual nosological categories of disorders might be represented in higher order factors. One factor, for example, representing internalizing disorders, another externalizing disorders. A more recent characterization shows that thought problems are also critical. Importantly, this thought problem issue seems to be the general vulnerability factor for internalizing disorders, externalizing disorders, as well as thought disorders. What we propose is that heart rate variability is an index of this general vulnerability factor. With respect to this factor, Caspi and colleagues note that adults with higher levels of P fared less well on tests requiring attention, concentration, mental control, visual perceptual speed, and visual motor coordination. Testing to the ecological validity of these deficits, people who knew them well said that individuals with high levels of P experience cognitive problems in their everyday lives. So these cognitive problems might be the source of this general vulnerability factor for psychopathology. In that regard, then, we have examined the relationship between heart rate variability and attention and mental control. And I'll just show you two sets of studies that characterize this. So in one study, we looked at the relationship between heart rate variability and intra-individual reaction time variability during an attention task. This intra-individual reaction time variability is thought to index a aspect of attentional control, such that rare, long reaction times may indicate attentional lapses. Now, we examined this in a modified flanker task where we had both congruent and incongruent trials in which an arrow indicated where a target might appear. If the target appeared where the arrow pointed, it was considered a congruent trial. If it appeared in the opposite, it was considered an incongruent trial. And we looked at the intra-individual reaction time variability. In this study, we assessed it using the standard deviation. And what we can see is that higher heart rate variability is associated with lower intra-individual reaction time variability as measured by the standard deviation of the reaction time for congruent, incongruent, and all trials considered together. Now, the standard deviation is a relatively crude measure of the intra-individual reaction time variability. This is just the scatter plot showing the inverse association between heart rate variability and intra-individual reaction time variability. Now, these distributions, however, may be characterized using ex-Gaussian function, exponential Gaussian function, the Gaussian part having two parameters, mu, the mean, and sigma, the variability, and the exponential part having a parameter tau, which represents the tail of these distributions. And these extra long reaction times are thought, as I indicated, to represent attentional lapses. So in a second study, we looked at the association between heart rate variability and these ex-Gaussian metrics using a similar type of task. And what we were able to show is that heart rate variability was inversely associated with tau in particular. Of our ex-Gaussian parameters, tau was the one that was most associated with heart rate variability. And this just shows the scatter plot of the association between heart rate variability and tau. Given that tau represents these attentional lapses, higher heart rate variability is associated with better attentional control, and according to the Caspi et al. Model, should therefore be associated with lower psychopathology. Now, cognitive control is important in a number of different disorders, and we published a paper on heart rate variability and cognitive control in post-traumatic stress disorder. But we further looked at heart rate variability in the context of control over memory retrieval. Given that intrusive memories are a problem for PTSD, the ability to control those memories might be particularly useful. So we looked at this using Michael Anderson's think-no-think paradigm. In the think-no-think paradigm, participants are trained on pairs of words and are given instructions during the task to either suppress or not think about the second word in the word pair, to respond, or given no instruction at all with respect to the word pairs. And then subsequently, they were tested using either the same probe, that is the same stem that was used in the word pair, or an independent probe based on a category that the target word might apply to. What we found was that indeed, as Michael Anderson has shown, individuals are able to not think certain thoughts or to suppress certain memories. Importantly, what we were able to show was that this was associated with heart rate variability, such that those individuals with higher resting heart rate variability were better able to suppress these memories, both using the independent probe and the same probe. This suggests that individuals with higher resting heart rate variability may be less susceptible to these memory intrusions that characterize disorders such as PTSD. We further examined the association between resting heart rate variability and this inhibitory control to assess whether this association occurred above and beyond impulsivity. And in this particular study with my colleagues in Italy, we used a rule shift cards task and a hailing sentence completion test, which are neuropsychological tests of inhibition. And what we found is that higher heart rate variability was associated with greater inhibitory control in both of these tasks. Now, what I've tried to show in this presentation is that heart rate variability may be important for psychotherapists. The pathway by which this might occur is via a P factor, a general psychopathology vulnerability, which may be indexed by heart rate variability. The basis of this general vulnerability factor being cognitive control issues, I tried to show the relationship between heart rate variability and cognitive control, and thus why psychotherapists might be interested in heart rate variability. So just by way of conclusion, just to show you again, pre-treatment vagal activity predicts treatment outcome such that those individuals with lower pre-treatment heart rate variability are more likely to show residual symptoms or to drop out compared to those who were fully recovered. And this was due primarily to those individuals who dropped out for motivational reasons, such as too much fear to enter the exposure situation. So I'd like to end with a quote from that is often attributed to Darwin, but was actually from Megginson. And he says that according to Darwin's Origin of Species, it is not the most intellectual of the species that survives. It is not the strongest that survives, but the species that survives is the one that is best able to adapt and adjust to the changing environment in which it finds itself. Persons suffering from psychiatric disorders are less able to adapt and adjust. And what I propose in this presentation is that heart rate variability may be an index of this ability to adjust and adapt to our changing environment. Thank you.
Welcome everyone, this is Sylvain Laborde speaking from the German Sports University Cologne, and I'm going to present you today a meta-analysis about the effects of slow breathing on heart rate variability. So this is an overview of the talk today. And as an introduction, some words about breathing techniques. Slow breathing has been used for centuries as part of meditation and relaxation techniques. One of the main mechanisms supposed and suggested to analyze these effects is the vagus nerve, the main nerve of the parasympathetic nervous system. And slow breathing is usually realized around 6 cycles per minute, which has been related to many benefits at the health and stress-related physiology levels. In comparison, spontaneous breathing lies between 12 and 20 cycles per minute. The effects of slow breathing are, for example, optimizing the functioning of the autonomic nervous system, optimizing the functioning of cardiopulmonary and neuroendocrine functions, and decreasing anxiety and arousal. It will also increase relaxation, and we also found some modest reductions in blood pressures documented in the literature. Talking about heart rate variability, the change in the time interval between two R peaks, we will focus in particular on two indicators in this meta-analysis, namely RMSSD in the time domain and low frequency in the frequency domain, when the breathing frequency is located below 9 cycles per minute. Some other indices will also be collected in the time domain, like SDNN, and in the frequency domain, so low frequency and high frequency parameters, calculated in terms of percentage, calculated in terms of total units, normalized units. And to summarize, the aim of this meta-analysis was to find out the effects of voluntary slow breathing on heart rate variability, considering 3 time points. First of all, we'll focus about the effects during slow breathing. This is to say, what happens when someone is doing slow breathing regarding heart rate variability. Then what happens immediately after one session. And finally, what happens after a series of sessions, after a long-term intervention, like let's say, for example, 4 to 10 weeks of slow breathing intervention. We followed the following PCOS parameters as inclusion criteria. We took both healthy humans and patients. We took all interventions with breathing frequencies below 10 cycles per minute. And as a control condition, we needed a breathing frequency higher than 10 cycles per minute. In terms of outcomes, we were interested in studies reporting HRV parameters. We pre-registered this meta-analysis on Prospera. And this is the different tools and method points that we use for the meta-analysis, also reported in the dedicated paper that we are currently preparing. If you want more details, you can ask me at the end. And we also run as a risk of bias, the risk of bias tool, ROB2, from Cochrane. In terms of statistical analysis, here's the display of different interpretations for HSG. And here's our results overview. In total, we included 223 studies. It's 172 during, so this is the most represented category. Then after one session, we only found 16, and after long-term intervention, 49. We also run, as I said, the risk of bias is just a short screenshot to show you what it looked like. Main thing to say about the risk of bias, most of the studies were reported as having a high risk of bias. I will come back to that in the discussion. Reading the results of our meta-analysis. Well, first of all, let's see what happens during. And indeed, during, we've got an increase of both parameters representing the vagus nerve activity, cardiac vagal activity, both for RMSSD, moderate effect, and for low frequency, a larger effect. After the session, these effects seem to remain, though quite small, here an effect size of 0.40. And after long-term interventions, also with RMSSD, we can also find a small effect, meaning that overall, now comes the discussion, we found this increase in parameters reflecting vagally mediated HRV in all the time points we were looking at. During the session, slow breathing session, immediately after the session, and also after long-term intervention. How can we interpret this? Well, to put it in a nutshell, as I said, one of the main mechanisms suggested to be responsible for the effects of slow breathing is the vagus nerve, which activity we can measure via heart rate variability in terms of its efferent activity on the heart, cardiac vagal activity. So slow breathing is likely to be a stimulation of vagal afferents that is then reflected in vagal efferents, going from the brain to the heart. And among the mechanisms linked to this vagus nerve stimulation, we can cite the baroreflex, the respiratory sinus arrhythmia, pulmonary afferents, and emotion regulation. Regarding RMSSD, we can say that even if we do find a small effect after one session, it does have the tendency to come back to baseline after some point. So let's say that effects tend to cease quite fast after the stimulation of slow breathing. But what is interesting is that on the long term, we do see some chronic adaptations which may reflect some chronic adaptations of the mechanisms we mentioned just before. In terms of the effects also after long-term interventions, it's usually linked to improve in cardiorespiratory function, improve in mood, and some decrease observed in blood pressure, perceived stress, and catecholamine and cortisol levels. So overall, this picture of having a relaxing effect on the body. As I mentioned, for the risk of bias, we do have a high number of studies with a high risk of bias, meaning that most studies did not report all HRV parameters. Most of all of the studies focusing on 1, 2 HRV parameters. Then the blinding of participants was not always possible, given slow breathing is kind of an obvious intervention. And also some issues leading to the randomization of participants. Future research agenda is directly linked to the methodological limitations we just mentioned. And in particular, we really recommend further research to fully report the HRV parameters. Given when you do HRV analysis, you basically get all the parameters. And if you don't have space in the paper itself, then you can do it, for example, as a supplementary material in most of the journals. Then also, please report a detailed methodological report of your study so that we can easily follow, so the reader can easily follow what was done and understand the intervention, so then we can slowly come to the standardization of slow breathing intervention. Reaching the conclusion, we can say that slow breathing triggers an increase of the parasympathetic nervous control of the heart, which really point towards a recommendation to implement slow breathing into health-related contexts. Still, keep in mind that the methodological quality of the studies should be improved. Thanks for your attention. I have a couple of reference slides, happy also to share. Just write me an email and I'd be happy now to take some questions. Thank you very much for your attention. Talk to you soon. Bye-bye.
First of all, I would like to thank Sonia and Pascal for organizing this interesting symposium, and I would like to thank you for inviting me. Thank you very much. I'm going to talk about perseverative cognition, and here are the main co-authors implicated in the work I'm going to present today. Perseverative cognition is a recognized risk factor for the onset, maintenance, and recurrence of several psychopathological conditions. It's implicated not only in mood and anxiety disorders, as initially thought, but also in disorders with dysfunction in the positive balance system domain, such as, for example, the manic phase of bipolar disorder or substance use disorder, up to the point that it's today a well-recognized transdiagnostic risk factor for psychological health. Not only it's implicated in psychological health, but also in somatic health. This is the core of the perseverative cognition hypothesis developed by Jos Broschut and colleagues, positing that perseverative cognition is capable of eliciting a physiological stress response similar to that evoked by actual stressors. And the term perseverative highlights the fact that such response is likely to be prolonged over time. And we all know that the stress response has evolved in the service of survival. It can save our life in the short term, but if prolonged, it heightens the risk of developing somatic diseases. To quantify the entity of such association between perseverative cognition and physiological activation, we conducted a meta-analysis of the literature, and indeed we found that perseverative cognition is associated with increased systolic and diastolic blood pressure, increased heart rate and cortisol, and decreased heart rate variability. And even if side effects are small to medium, there is room for larger effects if important moderators are taken into account, for example, if studies of better quality are conducted. Two key ingredients are important when studying perseverative cognition, that are flexibility and inhibition. Perseverative cognition represents the inability to inhibit a context-inappropriate response. And at the peripheral level, such inhibitory control relies on the vagus nerve that constantly inhibits sympathetic nervous system arousal. And at the central level, the prefrontal cortex is deputed to such inhibitory control. And in fact, a meta-analysis of neuroimaging studies by Julian Thayer and colleagues has found that heart rate variability, which is a proxy for vagal inhibitory control, is associated with prefrontal cortex inhibition, such that higher heart rate variability is associated with better prefrontal control over subcortical regions. In a study that we conducted on a sample that has perseverative cognition as a diagnostic criterion, that is the generalized anxiety disorder, we have found that patients with generalized anxiety disorder have reduced functional connectivity between the prefrontal cortex and the amygdala, likely meaning reduced inhibitory control in this patient and reduced ability to inhibit thoughts, maybe. This is accompanied by reduced heart rate variability. And the most interesting result of this study is that when we induce perseverative cognition in healthy control, this has the effect of reducing in functional connectivity between the prefrontal cortex, the intermedial prefrontal cortex, and the amygdala. So a perseverative cognition induction has the effect of making healthy individuals similar from a functional connectivity point of view to patients with generalized anxiety disorder. At an autonomic nervous system level, this is mirrored by a reduction in heart rate variability. We performed another meta-analysis of the literature to see whether if we can experimentally manipulate cortical excitability by the use of non-invasive brain stimulation techniques, then we can also have effects at a physiological level. And indeed, we have found that we can reduce heart rate and increase heart rate variability by increasing cortical excitability in the dorsolateral prefrontal cortex. So we performed a series of studies to see whether we could use transcranial direct stimulation and TMS to manipulate cortical excitability of the dorsolateral prefrontal cortex and influence the physiological consequences of perseverative cognition. In this study, we used the TDCS and we used the trier social stress to induce perseverative cognition. This is a typical social stress characterized by a social interview where the participants had to talk about how they behave in social situations in front of a very judgmental panel and then count backwards. So it's a very demanding and stressful task. And then there is a long recovery where participants are supposed to ruminate about their performance and the negative feedback they got. We had first a 24-hour assessment, cardiovascular assessment with heart rate monitor and the cortisol awakening response. So we recorded cortisol at awakening and 30 minutes after. Then participants came to the lab. We had ECG at baseline, then cortisol again. Then they underwent TDCS, so excitatory TDCS or sham. This is a between subject design. And we So they underwent it for 5 minutes before the Trier social test and for the entire duration of the stressor, for a total of 15 minutes of stimulation. Then during the recovery again, we assessed cortisol after 10 minutes and after 30 minutes. And again, they had a 24-hour ECG recording with cortisol awakening response the morning after the laboratory session. What we found is that a nodal TDCS, so excitatory TDCS, was capable of reducing state anxiety after the social stressor. So it favors the inhibition of the anxious response after stress exposure. At the physiological level, here you see change scores. SI is for social interview, AT is for the arithmetic task. And you can see that active TDCS compared to sham inhibits the physiological stress response during the task. And if you look at panel D, where you can see sympathovagal balance, so the ratio of low to high frequency, you can see that there is an asterisk even during the recovery phase, meaning that not only during the task they have a reduced physiological stress response, but also during the recovery phase, sympathovagal balance is better restored. So active stimulation, so increasing cortical excitability, this is what we have done in the dorsolateral prefrontal cortex, is capable of dampening the physiological stress response and also has effects in the recovery phase where participants are ruminating. We didn't find any effect on cortisol in the short term, meaning in the lab. There were no differences between the two conditions, but we found effects in the long term. So the cortisol awakening response was dampened in the group that underwent a sham stimulation, and this is a typical result in the literature. Stressor can interfere with the adaptive cortisol awakening response the morning after. So what the active stimulation did is to prevent the effects of acute stress exposure on cortisol awakening response. So even one session of stimulation seems to have prolonged effect on the physiological consequences of perseverative cognition. Work by Peggy Dockola has shown that perseverative cognition is associated with consequences on the cortisol awakening response the day after. Then we did a TMS study to see whether we could do the reverse, meaning if we could inhibit cortical excitability in the dorsolateral prefrontal cortex by the use of continuous theta burst stimulation. So what we did in this case is we used an inhibitory session with the hypothesis that this would increase the physiological stress response associated with perseverative cognition. This was a within subject design, and we also had an active control condition where we used the same stimulation protocol on the ventral premotor cortex, because the side effects are similar. The pain and the tingling are very similar for the dorsolateral and the ventral premotor cortex. And then we had the usual passive control condition, the sham condition. Here, so we used neuronavigation to make sure that we were stimulating the right side, the right region. We assessed each EKG at baseline, then we induced perseverative cognition as usual, asking participants to recall an event in the past that made them stressed, angry, and they keep thinking about it today, or something that worries them in the future. And then because participants had to come to the lab 3 times during the same afternoon, and we had a long washout period, we used prompt. So we didn't repeat the induction every time, but we used prompts, so words or sentences from the recall interview to trigger intrusive thoughts. And then, so they underwent stimulation, again, ventral premotor, sham, or dorsolateral prefrontal. Then they had a 2-minute rest to make sure that the stimulation was effective. Then the prompt to elicit perseverative cognition. And then they did a tracking task that we used many times. And it's very useful to assess perseverative cognition with a very low demanding task. Here you can see there is a circle moving very slowly on the screen of the computer. Every time the circle becomes red, as you see on the right, they have to press a button, and we record reaction time as a proxy of the ability to get off thoughts. And also there are thought probes asking about subjective focus. Are you completely off task or on task? And subjective threat. Are you completely safe or in danger? And what we found is that if we inhibit cortical excitability in the dorsolateral prefrontal cortex, we obtain increased heart rate compared to the sham and the ventral premotor conditions. We decrease heart rate variability, compared to the other 2 conditions. Cortisol is also higher for the dorsolateral prefrontal cortex inhibitory condition, but we didn't find any effects on the behavior, so reaction time, and on subjective reports. So what can we conclude? That the left dorsolateral prefrontal cortex is likely to play a major role in the physiological stress response that accompanies perseverative cognition, as if we increase cortical excitability, we have decreased physiological response, and if we inhibit cortical excitability, we have an increased physiological response. So adopting a causative approach, we were able to establish such a role for the left dorsolateral prefrontal cortex, and this is supported by other studies in the literature. Some of them did not find effects on cortisol, some did not replicate effects on heart rate variability, and this is very likely to depend on the type of protocol and the type of induction used to trigger perseverative cognition. So it would be very important for us to use the same induction, the same condition to better replicate our results. We didn't find any effects on self-report or reaction times, but this is not really surprising, as a meta-analysis of the literature found that non-invasive brain stimulation of the left dorsolateral prefrontal cortex does not influence self-reported emotional stress reactivity. And another study conducted in 2020 also found, like we did, physiological effects without concomitant subjective effects. However, TMS is used in the treatment of depression, and changes at the subjective level are crucial expected outcome of randomized trial. Of course, we did 1 session, whereas randomized trial have more than a session. I think that it's mandatory to establish dose-response relationship. So how many sessions do we need to elicit effects at the physiological level? What is the duration of such effects? It's very important that we establish dose-response relationship. Our studies have limitations. The first one had a small sample of young, healthy men. We were able to overcome some of the limitation in the second study, where we used a within subject design with also an active condition. but maybe the tracking task was too low demanding to show effects at behavioral level. Maybe one question was not enough to assess ongoing perseverative cognition, and this is why we didn't find effects at the subjective level. It would be important to have a deeper assessment of subjective perseverative cognition and mood during this type of studies. Also, it's crucial to include a right dorsolateral prefrontal cortex stimulation, as the lateralization model of autonomic control of the heart remains controversial. And we did the left because treatment for depression has the left dorsolateral prefrontal cortex as a target, but in the future, it's auspicable that future study will also stimulate the right dorsolateral prefrontal cortex, and hopefully combine non-invasive brain stimulation techniques with neuroimaging techniques to record the changes not only in the target region, but also in functional connected brain regions to elucidate the neural mechanism underlying the observed effects. And with this, I thank you for your attention. And I want to thank all of my collaborators in all of my studies, and especially my mentor, David Shapiro, who was a giant in psychophysiology and who passed away last year. Thank you very much. Bye-bye.
Welcome, everyone. Sephora Minjot speaking from the French Université Savoie-Mont-Blanc. So today I would like to present you one of my thesis projects concerning the interest of heart rate variability biofeedback method to restore neurovegetative disturbances and psychological distress in patients suffering from somatic disorders. So I'm going to start by conceptual introduction, and I will carry on with experimental design and method, following by the presentation of some preliminary results, and finish by some point of discussion. First of all, I will define the somatic symptom disorders as the main disease of interest of this project. So somatoform disorders are characterized by various physical symptoms, reminding us some somatic affection, but without any organic origin, and no physiological mechanism have been identified yet. This disease is mainly manifested by somatic, cognitive, emotional, and neurovegetative disturbances with a principal stress trouble. And in fact, a somatoform disorder is considered as a stress disorder. Stress makes symptomatology worse, and it could be a contributing factor in somatoform disorder occurrence. At the moment, the diagnosis of a somatoform disorder is based mainly on a psychiatric model in accordance with the biopsychosocial approach. And some researchers have recently defended the idea of common factors of vulnerability behind the large variability of the clinical symptoms regarding somatoform disorders. So in this context, the lead of the neurovegetative disturbances as autonomic nervous system disruptions started receiving attention following some studies that suggested a tonic dysautonomia concerning a somatoform disorder, and independently of its form. In addition of peripheral symptoms, we know that SD are classically characterized by a common central symptom, especially cognitive emotional distress, such as depression or anxiety that could be linked with autonomic dysregulation. In this way, even if resting heart rate variability reveals dysautonomia, it doesn't indicate the level of vagal flexibility under stress. And as I said earlier, if the somatoform disorder is a stress trouble, it may be worth investigating autonomic nervous system dynamical response in a stress situation. And both of the properties of cardiac vagal control, either tonic or phasic, are important to consider regarding adaptation abilities of the organism. But at the moment in the literature, there are rarely elements regarding this level of vagal flexibility. That's why we have decided to explore heart rate variability, reactivity to and recovery from stress in order to measure the level of vagal flexibility. And we assumed that as the patient would suffer from a dysfunctional autonomic flexibility in addition to tonic basal dysautonomy. This is how we were interested in two types of somatoform disorders. The irritable bowel syndrome, manifesting mainly by peripheral symptoms, and the psychogenic non-epileptic seizures, manifesting by central symptoms. So now, by knowing the physiological and psychological trends trouble in ASD, we have liked to restore them, and to do it we found interesting to use biofeedback method. And in this context, heart rate variability biofeedback has received several clinical and experimental confirmation as a physiological remediation method. So it could be a powerful method because of its positive impact on heart rate variability, and especially on vagal tone and baroreflex sensitivity related to the autonomic balance that could restore dysautonomia. And in fact, biofeedback is an innovative and non-pharmacological therapy frequently used to relieve stress. So taking in account the last arguments, we decided to evaluate the feasibility and efficacy of the biofeedback method. And according to the guideline, a fractional training is proposed, 5 minutes, 3 times a day for 24 days. So now let's turn to the protocol research. So I'm going to shortly describe the experimental design. So for the clinical trial, so we would like to include 25 patients suffering from an ABS and 25 patients suffering from a PNES. And at these times, 1 PNES is included and 7 IBS. For the research in healthy population, we would like to include 44 healthy subjects divided in 2 groups. And at this time, 8 are included in experimental group and 6 are included in the placebo group. Here's an experimental design of the clinical trial. So 3 sessions with 24 days between each. And it's important to mention that the first 24 days between the first and second session formed the control period, as a period without any training or exercise. And in contrast, the second 24 days between the second and the third session formed the intervention period, as a period included biofeedback daily trainings. Concerning the healthy subjects, so it's the same design, except the intervention period, which is divided in 2 sub-interventions. Some healthy participants were included in the experimental group and had to daily train biofeedback, like patients, and the other were included in the placebo group and had to daily train pseudo-biofeedback. Next, the session design for each session. So it was the same for all sessions. At first, participants have to complete questionnaires. After that, we are playing all electrodes. Then we start by a resting period, followed by the mental stress task. And the session ends with resting recordings in the same condition as a baseline period. Here are some physiological indicators measured in a study, and today I will present only RMSSD reactivity and recovery parameter referring to our vagal flexibility. Next, psychological indicators with different surveys, and today I will only present PANO score referring to positive affectivity. So now I will present some preliminary results concerning the physiological effect of biofeedback, and especially on RMSSD reactivity to and recovery from mental stress tasks. And we hoped for vagal flexibility in accordance with that I said earlier concerning stress issue in ASD. So first, no significant results are observed at this time. But by descriptive analysis, we can see on the first graph, we can see the RMSSD reactivity score referring to the vagal withdrawal through session. So session 1, session 2, and session 3. So in the first session, the reactivity score looks similar between all the group, and in contrary to our expectation, we didn't observe a specific phasic dysautonomia in ASD patients. Then, in accordance with some of our suggestions, there was no RMSSD change between session 1 and session 2. And regarding the third session, the results suggest no significant effect of biofeedback training on heart rate variability at this time, and illustrating by comparison no difference with a comparison between groups following the intervention period. Next, on the right graph, we can see the RMSSD recovery score referring to the vagal rebound through a session. In the first session, as observed for the reactivity score, the recovery score looks similar between all the groups, and we can't see a significant difference between healthy and patients. For the second session, as expected, there is no RMSSD change following the control period for both patients and placebo groups, but concerning the experimental group, we can observe a slight increase of the score. Now, regarding the third session, in agreement with our expectations, the recovery score had been modulated by the intervention, illustrating by a slight increase of the vagal ribbon in the LC experimental group using biofeedback training, in contrary to the placebo group, where we observed a decrease. But our results had to take carefully because we didn't observe this modulation in our SD patient group. Now, dealing with emotional effect of biofeedback, here is a score of positivity affectivity through the differentiation. So even if no significant results are observed at this time, descriptive analysis seems to confirm that patients suffering from ASD are likely to have a lower affectivity in session 1 in comparison with LC subject in favor of a higher emotional distress. And at the end of the biofeedback training, so if we see the third session, we can observe a slight improvement of affectivity, specifically for the SD patients group. So, so contrary to our supposition, first, we did not obtain significant effects of biofeedback, significant effect of biofeedback on the cardiac indicator, which does not seem to be in line with the vast majority of studies. However, when taking a closer look, there are a handful of studies that didn't obtain significant differences in these indicators. And in this context, our results are part of present debates about efficient and consensual biofeedback practice guideline that could be maximize heart rate variability effects. Additionally, recent work suggesting that biofeedback efficacy depends on the resting autonomic state, before starting the intervention. And some subjects look like a good responder, while others not. And this depending on their initial autonomic profile. Concerning our results, our small sample size did not allow us to cluster the participants on the basis of their autonomic profile, but it could be a track to understand the absence of physiological effect. Regarding our result on psychological indicators and considering that no significant physiological change are induced by the biofeedback, we can suppose that the method could have increased vagal afferences traffic, allowing a better integration of the internal state by the central autonomic network and allowing a better emotional regulation. And more recently, some authors suggested the resonant frequency focused on 0.1 Hertz could be a specific marker of the cognitive regulation of negative emotion. And the resonance bracing techniques could have had dominant and starting effect on emotion, so related to bottom-up processes, and effects on heart rate variability could have observed later as more deferred effects related to top-down processes, as efferences, and has supported by some recent research, which observed, in fact, physiological effects 3 months follow-up vagal nerve stimulation. So insert more long-term measurements could be interesting to a greater understanding of physiological effect. And nevertheless, we can also mention some strengths of this study. So first, we use the strict methodology in accordance with the latest guidelines concerning physiological measure, and we used strict inclusion criteria. We also try to verify the larger biofeedback effects than the placebo effects by comparing the biofeedback technique with a sham technique, the pseudo biofeedback. And we also try to verify the larger biofeedback effects than the unspecific effects related to the spontaneous variations through a control period. And based on the literature, in fact, the placebo effect is not commonly tested and represents one of the first limitations identified by some authors. We observed a good compliance capacity, and we can highlight the feasibility and acceptance of the protocol because all of included subjects have gone at the end of the protocol. Anyway, we can also stress some limits, such as the impossibility to counterbalance control and biofeedback condition, to use a double-blind protocol, or to control emotional events during the intervention period. To conclude, some of our preliminary results are promising, but will be needed to be confirmed by our total research on larger samples, which is in progress at this time, to assess the relative contribution of the biofeedback training. So thanks for your attention and enjoy the Congress.
Good morning or good evening, depending on your time zone and your personal preferences to watch pre-recorded videos. I'm Catherine Talon-Baudry, working in Paris. And today I'll present you some facts and open questions about gastric brain coupling. So when we think about the gut-brain axis, what comes immediately to mind is a microbiota. And while there are links between microbiota and behavior, some mediated by the most direct pathways through the vagus nerve, this is not the type of mechanism I'll be talking about today. I'll be talking about another type of gut-brain coupling, which is the coupling of the brain with the gastric rhythm. So you might not be very familiar with the fact that the GI tract, the gastrointestinal tract, is generating its own pacemaker electrical activity. I will detail that in a moment. But you all know, for instance, that the heart is producing its own electrical activity, and there are two-way communication between brain and heart. Well, something probably very similar is happening for the GI tract, and here in particular, the stomach. So why look at such interactions? One is very mechanistic. One can consider the brain as a complex system generating complex spontaneous dynamics. And the fact that there are oscillators attached to this complex system probably modifies the dynamics and should also help to better understand brain dynamics. But from a more biological and cognitive perspective, the brain and the intestinal organs are all part of the same organism, and it's important for this organism to remain well-fed and well-regulated, and this is done through interactions with the environment. So it might well be that the communication between brain and GI tract is also very much related to perception, action, and cognition. So let's start with some basic facts about the stomach. So the stomach is, of course, involved in digestion. It mixes and grinds food during digestion and ejects it in the intestine. And this is thanks to contractions of smooth muscles. So there are two rings of smooth muscles. But here I'm more interested into what's going on in the stomach outside digestion, and at all times during digestion, but also outside digestion. These cells here in blue are called interstitial cells of Cajal, generate a slow electrical rhythm called the gastric basal rhythm. And those cells are electrical pacemaker that generate this rhythm at all times, even outside digestion, and even when disconnected from the brain or when cultivated in a dish. So this is really an intrinsic rhythm. Those cells make contact with vagal sensory neurons, but they also make contact with the smooth muscles. And actually their primary role, as described in the literature, is that when there is food in the stomach, there is a reflex which increases the amplitude of the activity in those cells and entrains the smooth muscle contractions and organizing them so that it first presses at the top of the stomach to push the food downward rather than upwards. So it's relatively easy to record the gastric basal rhythm with electrodes placed over the abdomen, cutaneous electrodes, similar as used for the electrocardiogram, for instance. And here is an example of raw data in a participant from one of those electrodes, where you can see with the naked eye this very slow rhythm, which is a gastric rhythm. So one cycle is 20 seconds, that's super slow. You can see superimposed on it this faster rhythm, which is the respiratory rhythm, 4-5 seconds. And each of those vertical bars that you can see also in the inset here are actually heartbeats, so one every about 1 second. The gastric cycle is not always seen with the naked eye in all participants, but the spectral signature is almost always there, a very sharp spectral peak around 0.05 Hertz. So we wondered whether we could consider the stomach as an electrical pacemaker generating this very slow rhythm, and that this might constrain or at least explain part of spontaneous brain activity. So here we measured brain spontaneous activity using Fmri. So we measured the ball signal and we recorded the electrogastrogram at the same time. So if I represent the gastric rhythm here in blue, in brain activity, we've been searching for voxels whose activity was locked to the gastric rhythm. So it could be a voxel with a time course like this one in red, with almost no time lag with the stomach, or another one like this light orange one, where there would be a large time lag with the stomach. What really mattered was that this lag was constant over time. So here is what we found in 63 participants. I think the first striking finding is that there are a lot of voxels which are coupled to the stomach. Okay, what we call the gastric network, so those voxels that are significantly coupled with the stomach rhythm, the gastric network is extended. The second striking thing is that we find a lot in sensory regions. You can see almost the whole medial occipital lobe extending, well, the visual system, but also somatomotor regions, auditory regions, posterior insula. So to analyze this a bit more vigorously, here we superimpose the gastric network in orange with the 7 canonical resting state networks. Like you have here the visual network in purple, and we find that almost 30% of the gastric network is in the visual network, while almost 40% of the gastric network is in the somatomotor auditory network, here in light blue. Okay, so altogether, we have about 70% of the gastric network, which is in sensory and motor regions. And all primary sensory cortices are coupled, including the primary olfactory cortex, if we make a region of interest analysis this time. So what does this all mean? Well, what we have is something a bit like a wave, so that we have some voxels which are synchronized early in the cycle, whilst others are late in the cycle. And early regions are in particular in primary somatosensory cortex, while we would find late regions in the visual system. So it could be some form of scaffolding mechanism, a coupling mechanism between sensory and motor regions. But what is also striking is that it bypasses most of the control regions, except for some parts of the default network. There isn't much in the control regions in the frontoparietal network, for instance. And this is a bit in contrast with classical ideas about visual regions. So here is the now quite old view of mesolam of brain organization, where here in green, you have the visual system beginning with D1, and here in blue, the auditory system beginning with A1. And the idea was that all those informations would get more and more integrated into higher regions. So in mesolam view, that would have been heteromodal regions, paralimbic and limbic regions. So mostly regions that we find completely spared by the gastric network, but we do find the gastric network here and here. So it means that regions that are commonly viewed as mostly independent are actually coupled through synchronization with the stomach. And a more modern view, which is this very classical segmentation of the brain into 7 canonical resting state network, is also based on the idea that if you look at instantaneous connectivity, the visual regions are independent from the somatomotor auditory regions. But when we look at delayed connectivity, as we did with the stomach, now we find that there is coupling. So those so-called independent regions are actually coupled through their synchronization with the stomach. So we looked at the bold signal, which is a very compact and indirect measure of brain activity with very slow dynamics. And we wanted to test whether some specific brain rhythm would be coupled with the stomach. So this time we recorded spontaneous brain activity while participants were sitting in the magnetoencephalography scanner. measuring electrical brain activity through magnetoencephalography, and we recorded the electrogastrogram again. Here we've been looking for phase amplitude coupling, which is a very general rule of coupling that was initially identified in the group of Yeri Boujaki, where you would have a slow oscillation here in blue, and the phase of this oscillation, so in a way whether it's a trough or a peak, constrains the amplitude of higher frequency rhythm. So here in this example, you would have a large amplitude during trough, a small amplitude during peak, large amplitude during trough, etc. So this is phase amplitude coupling. It has been found initially in rats, but also in humans, within a region, between region, etc. So now we wanted to test whether this rule also applies when you consider a brain rhythm, so the low frequency oscillation, when it's generated outside the brain. And we indeed found significant phase amplitude coupling between the gastric rhythm, the phase of the gastric rhythm, and the amplitude of the alpha rhythm, here very specifically at 10 and 11 hertz. We analyzed all brain rhythm between 1 and 100 hertz, and there was a very specific effect in this frequency range. And you can see the posterior distribution of this effect, and we find that indeed most of the effect is explained by phase amplitude coupling in those regions along the parieto-occipital sulcus that are known to generate the alpha rhythm. And we could also show in this particular case, using transfer entropy, that the directionality of the interaction is really from stomach to brain, shown here, rather than the other way around. Okay. So we found that this influence from the stomach on the brain explained about 8% of alpha variance. So it's not like the phase of the stomach rhythm explains everything that's going on in the alpha range, which is good news, I would say. But on the other hand, it's not negligible. I mean, it's a share of alpha variance that might have consequences. So finally, we tested whether we could find a link between gastric brain coupling and anxiety. In English, one says that one has butterflies in one's stomach. In French, we say that we have a knot in the stomach when nervous and anxious. So we analyzed the link with anxiety scores and found actually evidence for no link. So we analyzed both the parameters of the electrogastrogram in a quite large sample of participants and in Fmri. So this said, we've been studying young healthy adults. So we had a fairly large range of anxiety scores, but not the super high scores, and we did not induce anxiety. So probably further studies are needed, but I mean, if there is a link, it's not such an obvious link. Okay, so to conclude, the gastric rhythm accounts for a non-negligible part of brain dynamics, both in fMRI and in MEG. This might represent a scaffolding mechanism by which sensory and motor regions would be coupled through the gastric rhythm, but this scaffolding mechanism would be independent from control regions. We don't know yet what the behavioral relevance of this coupling is, although I'm hoping to have more news fairly soon. And so far, we have evidence for an absence of link between gastric brain coupling on the one hand and anxiety, at least in young healthy participants at rest. So thank you all for watching this video. I'll be more than happy to answer live questions on September 9. And I want to say huge thanks to the PhD students and postdoctoral fellows who are in my group.
time to close our symposium session. We thank the speakers for the exciting presentation. In few words, we have seen today that the study of the interaction and communication between the viscera and the brain via the ANS is at the dawn of an important development for the understanding of emotion and their regulation. The field of application of visceral-brain interactions seems to be very wide in both physiological and pathological conditions. Thus, it requires the development of standard procedure in order to be able to establish comparison between studies. From this point of view, the etude of HIV and respiratory biofeedback have been clear examples presented today. At a theoretical point of view, this last decade, theoretical models have been convincing, and in this frame, Toyer's model is one of the most influential contributions to propose a global model to understand the place of vagus nerve in psychology studies. In the study of vagus nerve, most of work have been focused on cardiovascular system, as revealed by the excellent recent paper of Christina about the heart in the mind. But we also seen today, through the Catherine Talon-Baudouin talk, that it is also possible to reveal the gut in the brain. This original approach opens a new way to study the influence and the role of autonomic afferents, particularly those mediated by the vagal nerve on brain functioning. All of this is very exciting, to my point of view, and we hope promising for future work in the field of emotion. So we look forward to seeing you at the Q&A session. Have a good day.